What is genetic epidemiology? Well, quite simply, it's the study of genetic factors in relation to health and disease. And this is a little bit different, maybe an extension of the classic epidemiology or study of epidemics. And in this module, we're going to evaluate how genes and environment may interact with each other in terms of producing a particular outcome related to substance abuse. We're also going to show how epidemiology, genetic epidemiology, really seeks to use or derive statistical and quantitative analyses of how genes may work in very large groups. The history of genetic epidemiology is quite interesting. It really started in the 19th century when addiction was considered a hereditary disorder. Now, one of the things for us to remember is that many things are hereditary, such as language. A person speaking French typically learns that from their parents, or religion. A person may be born into a family that is Catholic, but neither of these particular traits are considered genetic in origin. So one of the things we have to continually remember is when we're talking about genes and particular cause, whether it's an association or in fact there is a real effect of genes on particular traits or on behavior. We also need to begin to think about how genetic epidemiology in relation to substance abuse or disorders came about. And this really had its origin in studying the offspring of a parent, whether it be the father or the mother, and looking at the outcome of that individual. It's been known for some time that alcoholism and alcohol dependence, for example, do run in families. That is, if the parent has the condition of alcohol dependence, there is a very increased likelihood that one or more of the offspring will also develop the disorder. Back many years ago in the 1800s, and sometimes even as late as into the 1900s, persons with alcohol dependence or other psychiatric disorders were sterilized, often by a court order, in order to prevent the transmission further within a particular family. We began to understand more about the genetics in the 1940s as psychiatrists began to study twins in particular who were separated at birth. And with this separation, it was a natural experiment, as many other types of designs are, so that we could begin to separate the effect of genes from environment. If there was a genetic effect, we would expect the twins separated from the family to have the same risk of developing substance abuse disorders as those that were separated at birth or as their siblings who in fact lived in the family. If there was an environmental effect and we placed that individual in a good home, if you would, one in which there was no alcohol problems or other psychiatric conditions, we would then expect then that the risk would be significantly reduced. Currently, we think of substance use disorders really as a complex disorder or as a complex trait. And by that, what we mean are there are many pathways into the disorder. It could be from stress, it could be from other environmental influences, or it could be, if you would, a genetic predisposition. And so when we look at individuals who have alcohol or other substance abuse problems, what we see is really a variety of different psychiatric symptoms. And we've made a number of efforts over the years to try and homogenize these into different subtypes and used a variety of different techniques to do that. In terms of looking at the epidemiology and going forward. What we're trying to do when we try and homogenize, if you would, the heterogeneity of symptoms within groups of patients is to look at different trajectories of development from the onset into across the lifespan. And as we look at this disorders, one of the things we find is, in fact, there are many different pathways that lead to the development of psychiatric disorders. And unlike things such as hair color, eye color, maybe even height, most of the psychiatric disorders and substance use disorders really are polygenic. That is, there are many different genes that are likely to be involved in the development of a disorder, and many of these genes, in fact, contribute only a small amount of variance to the vulnerability, unlike some conditions that are more monogenic, where a single gene may in fact produce the disorder. It's also very well known in psychiatric epidemiology that there's a real interplay between environmental factors, things that happen in the environment, whether it's where the person lives, what they eat, the parental rearing patterns, etc., that may affect the genetic vulnerability and produce a variety of different outcomes. I like this cartoon because it really does demonstrate, I think, very clearly and with a grain of humor, that there is an interplay between genes and environment on a variety of different traits. In the first panel, in the upper left, what you see is two sisters talking about looking at their father and grandfather sitting out on the deck. And they note the physical similarity in terms of their nose, the mouth, the voice similarities, etc. And then also the behavioral part, in which how they look and are leaning against the railing in much the same way. And then the comment from the older sibling to her sister is, "Isn't it cool that two people of such different ages could be so much alike?" And in the panel, you see the younger sister mimicking exactly what her older sister is doing. Not at all uncommon in sibships. But let's take a look quick and see, get a better understanding of what we're talking about in terms of genes. In this particular slide, we are looking at yeast, which is very common in many foods that we eat, as well as the beer that we drink. Fruit flies, which are also very common, and particularly around fresh fruit. C. elegans, which are worms. Thale cress, which is a salad. And then rice, which is probably one of the more common foods eaten in the world. And as you can see there are a variety of, their genomes have been mapped, and we now know the number of genes in each of these different substances. But what do you think in terms of humans? Where do humans lie? When I started this work, it was quite given that there were probably 100,000 genes in the human genome. And as technology increased, we found that, in fact, that's not really quite true, that probably there are only about 20,000 genes in the human genome. So as you can see, humans lie just above worms and fruit flies and a little bit below the salad that you eat in terms of thale crest. So it's kind of interesting to note that the human genome, while quite large and quite complex, is in fact somewhat less than that of the rice that we eat.